name is Carl Peters. I'm an application scientist with BMG LABTECH, and thank you for coming. And I'm really glad that you're here to hear about some work that has been done with a collaboration with AssayQuant here in the United States. So my job is to introduce some of the reader technology that's employed that is capable of reading this very useful approach for monitoring and assessing the mechanism of action of protein kinases and phosphatases. The first instrument that I want to tell you about is our Fairstar FSX. And as many of you probably know, this is our high throughput screening type of instrument. Basically the best available technology for screening. And that this is a multi-mode reader, capable of very fast reads in all plate formats, even up to 3, 4, 5, 6 well plates. With this instrument, we've really stressed using dedicated technologies for the best performance. And this includes a xenon flash lamp that can be used for fluorescence or time-resolved fluorescence. So all of our all of the read modes in the Fairstar are based on an Optic module concept that is a self-contained piece of equipment that is easily exchanged within the reader, but has all of the elements that you need: excitation filters, emission filters, and dichroic built in. These are barcoded so that they can be scanned by the instrument, so that the software is updated and you can see exactly what read modes are available at any given time. So this reader has the best sensitivity and speed on the market, and our recent update a couple of years ago to the Fairstar FSX has seen even improved sensitivity in all the read modes, including 9 order of dynamic range. And of course, with the Fairstar FSX, simultaneous dual emission is always a possibility in case you're doing anything that has two outputs at once: fluorescence polarization, time-resolved FRET or FRET. A quite unique feature of the Fairstar line of readers is the ability to do decay curve monitoring. This is quite useful if you're trying to optimize an experiment with a time-resolved compound, be it any of the available kits or something that you've designed yourself. So this really helps you to optimize the settings in terms of delay and integration for those types of reads. The other reader that I'll tell you about as well is the Clara Star Plus, and these two systems really have shared a lot of different capabilities, and that includes the ability to perform top and bottom readings in all read modes, and to be able to perform a focal height adjustment and scan in both those directions as well. This is something that we have found is really useful for getting the maximum out of your assays in terms of the measurement intensity that you can achieve. And both of these readers can have a stacker for continuous loading. So if you're increasing your throughput, that's sort of the first step in the process. Some other things that are useful in both the Clara Star and Fairstar for the work that we'll be talking about today. It has a high-frequency flash lamp, so it can do kinetic reads very quickly. And on the data analysis side, it can be actually used to determine the KD values and perform the appropriate fits to be able to analyze the mechanism of action of the kinase. So now I'll move on to the Clara Star Plus, which has a couple of other features that will be useful. Again, this is a multi-mode reader. It's more used in the assay development side of things amongst our users. And that's because of its the flexible nature of detection that it can perform. And so again, it has the ability to perform all read modes, including fluorescence intensity and time-resolved fluorescence. And some of the features that have been improved recently are in the Clara Star Plus are enhanced dynamic range, which is going to be very useful, especially in kinetic types of assays. Once again, what we are stressing that each detection mode requires specialties for its particular detection, so we make sure that we're using dedicated detectors for all read modes. And both the Clara Star and Fairstar can have integrated barcode readers. Again, as you're increasing your throughput, that's a very, very useful way of tracking your experiments. So the main feature of the Clara Star is its linear variable filter monochromator. It's a patented technology. Unlike grading-based monochromators, it creates a filter-like performance, so that's why it has such high sensitivity. It also has scanning capabilities for when you are developing a new assay or characterizing a new fluorophore that you haven't used before. But when you need the best possible detection, filter-based detection is possible, and these are interchangeable. So you can excite with a filter and collect spectrum with the monochromator if that's what you need. So the key feature, as I mentioned before, is the enhanced dynamic range. And this does really two things. It expands the signals that can be detected in one run, so less chance of an overflow reading. And you can see both low and very high signals in one reading. And this has a secondary benefit of not needing to optimize the detection settings. So setting gain has always been a pinch point for a lot of users, and this eliminates that need. And that's especially useful in kinetic type of assays where you don't know for sure how quickly or how high the signal will increase. And so just kind of an illustration of what that enhanced dynamic range looks like. With manual gain settings, you could get only a few decades of detection. It increases the decades of detection that you can achieve in a single experiment, and it eliminates the need to adjust the gain these multiple times throughout the reading. And the reason that it's useful in especially kinetic types of assays, without enhanced dynamic range, you can quickly reach a saturation of your signal. But with the enhanced dynamic range, you are able to make adjustments and decrease the gain so that you can continually collect data that is useful to your determinations. It also has a redshifted PMT, and that's going to be useful for detection of all types of different fluorophores, including lanthanides, and that's relevant for the redshifted assay that Erik's going to be talking about a little bit later on. So with that, I will pass it on.
So we're going to tell you about some interesting work that we've done then from AssayQuant. Certainly in terms of protein kinases, and that's what I'll focus on today, both protein kinases and phosphatases. Very exciting area. And it's really driven by two market drivers. So one, the pharmaceutical industry's pursuit of blockbuster drugs, where it's estimated that 30% of all drug development is currently focused on kinases at this point. Phosphatases are always the sort of second cousin, but we're certainly seeing increases there as well. If you look at overall deals in the pharma industry, almost 40% of the value of all pharma deals in the last decade has focused on kinase inhibitors. Secondly, the opportunities then in disease diagnosis and the demand for personalized medicine really pushing forward then in kinases both in terms of targets but also in terms of readouts to understand signaling pathways. So just as an example, this Fierce Pharma article where it said, when it comes to cancer drug deals, one of the hot protein families is raking in billions. And it's really true. If you look at the IPOs and Series A, B, and Cs that have been done recently, as well as acquisitions where kinase inhibitors have played a critical role. So it's important, though, to point out that legacy kinase assay tools really haven't evolved that much, and they are generally inefficient, costly, or both. So this really limits patient and shareholder value, and current drug development pipelines then for kinase-targeted therapies are really handicapped. And you can see this just by looking at a few salient facts, which is $2.6 billion then is the average cost to develop a new drug. Only 3.4% of clinical trials succeed, and the whole process takes about 10 years. Drug discovery then is really constrained both in terms of scale and speed, and a lot of this is due to the lack of suitable tools for effective development of new kinase drugs. Finally, only 15% of the 518 known kinases have been exploited, and again, assay limitations are a major factor. So what do we need? We need better tools, and the better tools then will accelerate drug development, reduce costs, and improve efficacy. And importantly, these new tools must allow researchers then to identify promising lead compounds from the thousands to millions of drug candidates. You need to be able to conduct the specific measurements that reliably predict drug efficacy. And third, you need to enable then rigorous testing at all stages of the drug development process, as outlined there. So this is really what motivated Barbara Imperiali and myself to found AssayQuant in 2015. Barbara is a well-known professor of biology and chemistry, really has been a pioneer in the chemical biology space. She's a member of the National Academy of Science, and she is the inventor then of the SOX technologies that I'll talk about today. Myself, I bring 20 years of experience then working in the life science tool market, developing innovative assays, especially for protein kinases. So together, we're really committed to developing innovative new products to enable next-gen drug discovery. So outlined here then is the SOX technology, and I'll focus on this today. We have a number of different technologies, but this has been the focus since founding AssayQuant. So as shown here, in each of the scenarios, what you start out with is a SOX peptide. This C-SOX peptide is actually using the SOX moiety that is covalently attached to a cysteine moiety residue in the peptide substrate, and you position this then upstream or downstream of the potential phosphorylation site of interest, can be serine, threonine, or tyrosine. So that when you take this peptide substrate, you incubate it with the kinase, ATP, and magnesium, you get phosphorylation of the substrate at the site of interest, and this immediately then results in a 2 to 10-fold increase in fluorescence that you can then monitor in real time. So the real time allows you to look at initial rates of the enzyme. It allows you to look then at drug interactions with the enzyme and get a lot of really useful information ranging from IC50s to, if you're looking at covalence, K-inact over Ki measurements, residence time, and so forth. So it's really one of the key buttons is to understand the time-dependent nature of inhibitor action on its target. So the whole system then is based on chelation-enhanced fluorescence, again, using this sulfamidoxine SOX chromophore. As I mentioned, SOX is covalently attached to the cysteine residue, allows variation in N and C terminal flanking sequence on either side so that you can get the best substrate for a particular kinase. The assay is direct, homogeneous, and kinetic, i.e. continuous. Phosphorylation of the sensor, as I said, results in an immediate 2 to 10-fold increase in signal, and it's really ideal then for robust quantitative analysis, inhibitor potency, and mode of action. So early on, Barbara spent a fair amount of effort because we often had requests then from HDS or SAR groups for developing a redshifted version of the SOX fluorophore. And when you do this, you almost invariably end up with a much larger fluorophore. And for many reasons, obviously you don't want that. You don't want to interfere with the function of the enzyme or the inhibitor. So it turns out actually to be rather simple. So for europium assays, you need a chelate. And it turns out that the SOX fluorophore actually acts as a very nice chelate, so that all you have to do at the endpoint of an assay is to add europium. You can neutralize and stop or not. And you then just read at 360 nanometers excitation and an emission of 616 to 620. So you're still using a very small fluorophore, but now you're getting a redshift readout. And you're also getting time-resolved fluorescence, so you can then code in a certain microsecond delay, so that together you really eliminate then any interference due to compound autofluorescence. So as shown here, it uses the same optimized C-SOX peptide substrates, so you can go from HDS, SAR, to then doing mechanism of action, potency assessments, and so forth. The SOX, again, sensitizes the long-lived europium luminescence to give you that redshift readout with the 100 microsecond delay that's typically then used with europium. So here's then just the workflow, very simple. You can run these assays then in any plate format. We have groups then miniaturizing down to 2 microliters in 1536 well plates, so it ends up being very economical, as well as high throughput. You can read then in any fluorescence microplate reader. We've worked extensively now with BMG, and the quality of the instruments is really top-notch. So these are top-rate, next-generation instruments that give you much greater sensitivity than the earlier models. As I've said then, with a given sensor, you can do both kinetic, so it's a 485 emission readout, 485 to 505, or you can do an endpoint red, which is an emission of 610 to 620. And you can apply this both to protein kinases as well as protein phosphatases. So importantly then, with the enzyme kinetics then, you get all of the parameters for looking at enzymology, for looking at drug potency. But you can also, with the appropriately selective sensors, and I'll tell you a little bit about that, you're able to then look at unfractionated lysates from either cells or tissues. So really a virtue of the platform then is that you get direct readouts in a variety of samples, so that you can really cover the spectrum then of HDS, SAR, profiling, mechanisms of action, and potency. And so it really is simple, powerful, and flexible. And that allows you then to cover all aspects of the drug development workflow. And so we have groups then working with us early on as part of assay development to put in place the appropriate sensors. If they know they want to look at crude cell lysates, we'll do that right up front. You can then take those sensors all the way through the drug development process instead of developing multiple assays and having then potential conflicts between different assay formats. So it saves time, it saves money, it really speeds the process of drug development. But being able to look kinetically as a continuous format also allows you to develop better drugs. And so we have the 3 different formats, all can be using the same sensor. So it's really accelerating discovery and next generation drug development. And we have groups then already planning to take the crude cell lysate sensors where you have selective readouts for a given kinase or phosphatase and be able to use those as biomarkers as part of your clinical trials. And this is a real valuable readout to start to get to personalized medicine, start to understand the readouts of different pathways that will determine drug efficacy. So just to show you that as an example, if we look at staurosporin as a commonly used reference compound, we do IC50s then in the different formats. Shown first, the kinetic format, where you're determining reaction rates for every drug concentration. Or one can do it with an endpoint format, where you run your reaction, you then add europium either after stopping the reaction or not stopping the reaction. And in all cases, you get the same readout. In this case, then, it's low nanomolar potency. And what we see is that the values that we generate then looking at potency, either IC50s or more complex measurements of K-inact over Ki with covalent compounds, or looking at residence time, we see values then very similar to those reported in the literature or with other platforms. But the ease of use of the PhosphoSense platform really gives you much greater throughput, much lower costs, and ability to get to better drugs faster. So a critical part, if you think about protein kinases, a lot of the substrates that have been used have been around for decades. And they really, they haven't changed, they're not optimal. In many cases, they may be derivatized in various ways, adding lysines or arginines to help them bind to P81 paper or other types of modifications, whereby the resulting sequence is just suboptimal. And we've initially made many of these as SOX-based substrates, and they're quite lousy. So we then launched a major effort whereby we integrated the next-gen cysteine SOX technology that Barbara had developed to create a campaign where we would go after optimized substrates. So we created an FMOX cysteine SOX building block. We integrated this as part of high-throughput C-SOX peptide synthesis, so that we could make thousands of sensors from physiological sites reported for various proteins, and ask how do they work then as C-SOX sensors. And there are, of course, dramatic differences. So we currently have over 20,000 sensors, and I think what it really has highlighted is the clear opportunity to create improved and more physiologically relevant peptide substrates for use in our PhosphoSense platform. So we can target then a particular branch of the kinome, and with our 20,000 sensors, we have groups of sensors for every branch of the kinome. But here are just tyrosine kinases, which are obviously a very hot area. So we can generate then sensors that are either really generic, so they work with 80 to 90 different tyrosine kinases. We've had a lot of groups say, we'd like to have a relatively small number to cover all of our kinome assay set. But as I mentioned, we also can go after selective sensors, and we're increasingly getting requests then for this to go on to more complex samples. And here's an example where with one particular screen that we did, we could pull out a selective sensor for KDR, or VEGF receptor 2, and this is only a 9-mer. So it doesn't have to be a complex sequence to give you selectivity for the kinase of interest. We've had many groups saying you're not going to find many sequences that are going to be selective for an individual kinase or phosphatase, and I can tell you we're absolutely finding them left and right. And so it's really important to understand what is physiologically relevant, and are there mechanisms in place by which these kinases and phosphatases signal in a very selective manner, and the answer is yes. So we can target in on a hot area like the EGF receptor, and we have many groups coming to us saying that the EGF receptor substrates that are available are generally not very robust assays. We also had groups saying that, with all of the mutants that are being identified, there are clear differences in substrate specificity. So one of the requests was to come up with a much more robust assay that would work then across different mutants so you could characterize compound potency. So here's an example then where what we've done is done a couple of different screens. The first one looks at an individual substrate that we identified out of our 20,000 sequence panel. It works, but doesn't work so great. And we actually then go in and make individual substitutions at each position within this peptide substrate. And every one of these lines then is a progress curve that differs only by one amino acid in the sequence. So you see really dramatic changes with individual substitutions. We can then look at combinations of favorable substitutions and get to much better substrates. And so the goal is to make the minimum number of changes that give you a much more robust assay. And in many cases, it's only 1 or 2 changes out of a sequence of 20 amino acids. So it's a very powerful approach, and it's the real-time element of this PhosphoSense platform that just allows you to see it immediately. That is, that you see the improvements. So we can then look at a variety of different mutants. Right here is the wild-type EGF receptor and several then catalytically activated mutants. We get really high reaction rates, and this then translates into highly sensitive assays. So for the EGF receptor, we can go down to 160 picomolar of enzyme. For example, with the double mutant, what we see is 39 picomolar sensitivity. So just as what Carl was pointing out, everything is getting more and more sensitive. The drugs are getting more potent. You need to be able to drop your enzyme concentration so that you're at least twofold below the IC50. So we have a lot of groups coming to us saying that our assays are bottomed out. Our compounds are much more potent. We're in tight binding conditions, right? Can you help us actually get our assay back? And using this approach, this is exactly what we've done again and again. So we can look then with a reference compound, in this case looking at Tarceva. We can look at increasing concentrations then of inhibitor, and what you see for each compound concentration is a really nice linear progress curve that allows you to determine the reaction rate for that concentration of inhibitor with your enzyme. You can then plot that as a reaction rate, or in this case, looking at percent untreated control. So we can show you the wild-type EGF receptor versus the double mutant, where you have almost a 400-fold increase in resistance, so lower sensitivity. And that's exactly what you see in the clinic. So what you get then is a really accurate and precise assessment of inhibitor potency. And this is just one type of format. And what we've done is, in fact, develop then multiple formats. So using the PhosphoSense platform, we can look at pre-incubation effect of compound with enzyme. Some have really fast rates of inactivation. We can look at, with covalent compounds, we can look at K-ops versus I to get K-inact over Ki. We can determine whether it's a one-step or a two-step process. We can do then really nice correlations then of log IC50 versus the log K-inact over Ki. And this is actively then helping to direct SAR programs, I can tell you, with multiple companies. We can also look at residence time, here in blue showing the no inhibitor, here in green showing a jump dilution experiment where you pre-incubate the enzyme with an appropriate amount of kinase inhibitor. You then do a large dilution and you ask, what is the time required for the enzyme to regain activity? So we can see really dramatic differences between compounds that have very similar IC50s. We can see this either with K-inact over Ki's. We can also see it with residence time. So it's really helping then investigators make decisions as to which drugs to bring to the clinic. AstraZeneca is a group that has published with our platform. They have applied this to many different targets and have used high throughput assays then to identify different mechanism of action and desired potencies out of the thousands to millions of compounds. So they've moved the whole kinetic assessment much earlier in the process, directly after HTS. And just as I've said, with the red format that we have, you can actually use the same sensor to do the HTS screen, take them right on then all of your lead compounds to kinetic analysis and differentiate which compounds then you want to bring forward. And here's just an example where they've done high throughput, 384 well jump dilution experiments to look at a whole range of compounds with dramatically different residence time differences. So very powerful. So, obviously when we first started AssayQuant, there was a lot of interest, most of the interest, I think, around oncology. But that really has diversified a lot. So we see as many requests for inflammation now. And with some of the JAK inhibitors, AbbVie introducing their next-gen Humira, which is a small molecule JAK1 kinase inhibitor. So lots of targets. They tend to be much more difficult to develop assays for. Many of these are in complexes. And so we've spent a fair amount of time working out how to actually do this. So same approach, but just some different twists that I can get in as part of the questions. But here's just a series of assays that we've just recently developed for a variety of kinases involved in inflammation or immune cell signaling. So things like MAP3Ks have always been very difficult assays where it's typically been protein substrates that are used. And we can get really robust assays using peptide substrates, CSOX peptide substrates isolated in the way that I've mentioned. All of these are tough targets. If you look at the kinase suppliers, they're often using MBP or they're using an ELISA. So we've really cracked how to get then really robust drug development assays for these targets. And some of them, like PKR, or the RNA-dependent protein kinase, you're really interested in induction, in this case, looking at RNA. So we've applied the same approach to a variety of kinases that are activated by various regulators. And if we go through our CSOX libraries, we can find substrates that actually show very little activation and others that show very strong activation. And just some of the examples, AMPK, DNA PK, PKCs, we can look at a variety of lipid or calcium dependent activation mechanisms. I mentioned BKR, here CAMK, where with the optimized sequence as a substrate, you get dramatic induction then of the activity of the particular kinase of interest with its regulator. So in this session, I just want to touch on what we're doing with phosphatases. So again, it's sort of the cousin that doesn't get a lot of attention, but we are absolutely seeing a lot more requests for phosphatase assays, and what we can do is take a phosphopeptide CSOX substrate that gives you a high signal. You incubate that then with the appropriate phosphatase, and you see a time-dependent reduction in the signal. Same thing you can apply then to look at initial reaction rates. It's the same mechanism for chelation-enhanced fluorescence based on magnesium, kinetic assays, or you can apply the time-resolved fluorescence assays using the phosphosense red format. But importantly, all of our assays then use physiologically relevant CSOX-based phosphopeptides. And what groups are telling us that with a small molecule substrate like PMPP, you don't see a lot of differentiation between allosteric or substrate competitive inhibitors, but using the physiologically relevant phosphopeptides, you absolutely do. So we can incubate then a particular sensor then with increasing amounts of enzyme, and you just see dramatic then progress curves showing dephosphorylation of the site of interest. And these assays are incredibly sensitive. So we're going down to 20 to or so picomole of enzyme. And again, just highlighting that the reaction rates are so much more robust than what we even see with the kinases. And again, it's by virtue of the substrates that we've identified, again, being physiologically relevant. So we have then a growing panel of phosphatase assays, here showing SHP1-2, PTPN2, PP2A, calcineurin, and FLIP phosphatase 1 and 2, which is the enzyme that dephosphorylates AKT. So same approach, we can really identify very quickly optimized sensors. And here's just showing you an example of a phosphosense red format. So here the signal with AQT264, which is the phosphopeptide optimized for PTPN2, you see a nice strong signal. If you use the corresponding dephosphorylated version of that sensor, obviously you see a very low signal, and you can then add enzyme to the substrate sensor and get complete dephosphorylation of that sensor. Signal to background here is 7. The Z prime is 0.94. So really robust, excellent assays. So in the last few minutes, I've mentioned that we can clearly get selective sensors for individual enzymes so that one can start to interrogate then signaling pathways in crude samples. And Barbara then published a series of papers showing this approach using either cells or tissues, and she used matched tissue samples from the National Tissue Database, such that you make lysates of either of these. Again, you're doing this all in the same high throughput microplate reader format, but you're getting really quantitative readouts then of individual signaling pathways. You can see differences between cancerous and normal tissue in this case, and you can identify with a panel of sensors which then targets are being upregulated in disease, and subsequently show that those signals then are reduced with drug treatment. So as I mentioned, we can generate very selective sensors for using simple peptides, but there are other approaches where the enzymes, their specificity is more complex. And so one of the things that Barbara demonstrated was to show that you could combine various docking motifs with CSOC's peptide sensing sequences with a variable linker, and she did this for the MAP kinase superfamily, showing that you get really low KMs in each case with the selective sensor, and that they absolutely differentiate individual subfamilies, looking at either JNKs, ERK1/2, or P38. And so one can then look at, for example here, time courses, looking at stimulation of cells, in this case with a stress stimulus, endomycin. You see time course of activation for JNKs, ERK1/2, P38, and you can see the error bars that are all really tight because every signal is an initial reaction rate. So in this case, you actually get the similar signals, but it's much less quantitative if you use antibodies to the activation loop of individual MAP kinase members. But importantly, there are now, we know, 50 phosphorylation sites on each one of these kinases. And so the question is, some are positive, some are negative. How do you differentiate that? What's the algorithm that you use to really know that those enzymes are activated if you're using phosphospecific antibodies? And our point is just measure the activity directly. It's actually much easier, much faster, much less expensive, and much more informative. And here just one other example where Barbara looked at MCF10 cells at various stages of oncogenic progression. And what you see is in the highly malignant 100% xenograph efficiency cell strain, you see dramatic upregulation of the P38 MAP kinase and its downstream target, MAPCAP kinase 2. So you can just very quickly pinpoint which pathways are dysregulated. And this we're seeing is really being valued. We had a meeting with Neil Rosen just last month where he's very interested in looking at biopsies from cancer patients where they've been treated with various drugs to understand which pathways are contributing to resistance. So I think this really is a future where our platform can help enable these measurements. So as a summary slide then, the PhosphoSense platform delivers many advantages. It's simple, one-step add and read, continuous, gives you more information with less effort. You set up your assays, you see it immediately on the screen, but you can walk away and do something else. The system is optimized both in terms of the detection chemistry as well as the peptide substrate sequence that I've highlighted throughout the talk. The fluorescence signal is direct, it's quantitative. You can even convert to micromolar per minute in terms of your reaction rate, so you know exactly then what amount of substrate is being phosphorylated. It's predictable development in terms of robust assays, even with difficult targets. And so we have many groups coming to us and we will develop assays. We have very rapid turnaround to make the technology and these assays available. And get high accuracy, precision, and great lot-to-lot consistency. So in terms of versatility, you can use continuous kinetic fluorescence intensity or the endpoint red TRF format with the same sensor. You can use any ATP concentration, and as I've shown you, a variety of different sample types. So you can use these sensors across the entire drug development workflow. They run on commonly available plate readers in a variety of different plate formats so that you really get higher throughput. And just as we've talked about, the new instruments really are giving much higher sensitivity, further empowering then your detection capabilities. Finally, then we're expanding rapidly products and services. We have an increasing number of catalog assays, now 262 kinases, 8 phosphatases. I put some lists on the back that might be useful for you to look through. We also offer then custom assay development services and custom testing services where we have groups sending us compounds and very rapidly then showing how we can differentiate different compounds as their lead drug development efforts. Finally, then thank you for your interest. I would certainly take any questions. I also have a poster today at number 1283 from 5 to 6. So I would welcome any questions here or there and look forward to talking with you. Thank you.